Biochemistry of AIDS and HIV transmission: about 80% by sexually transmitted diseases (STDs), about 15% by blood, about 5% by mother to fetus. Approx 30% of infants born to HIV mother may get the infection. Course: window period: when virus enters the body, it is multiplied in body cells, but it cannot be detected easily. The virus capsid antigen P24 can be detected in blood. Seropositive stage: few months later, antibodies are seen in circulation. This is seropositivity. During this period, person is normal but a carrier. About 10% of seropositive individual progresses to third stage within 5 years, about 50% within 10 years, about 90% within 15 years if not treated. AIDS disease. Third stage is when the clinical manifestations started. By this time, immune system depleted. Commensal microbes increases. Patients die within 2 years after third stage. Some infections by fungi, candida and cryptococcus viruses, cytomegalovirus and herpes simplex virus. Parasites, cystic carinae and bacteria, mycobacterium and salmonella. Cancer associated with AIDS are Kaposi sarcoma and NHL, non-Hodgkin's lymphomas. Congestive impairment due to meningitis, neuropathies and neuropsychiatrist manifestations. Clinical manifestations: lymphadenopathy and fever may be seen by end of second stage. AIDS related symptoms ARS are wide. Since immunity is decreased, non-pathogenic microorganisms enter into the body and produces lesion in skin, GIT, lungs, urinary tract, brain, etc. Gastroenteritis and tuberculosis are predominant in AIDS in India. Laboratory analysis: by ELISA test, Western blot confirmatory T helper count smaller than 300 cubic mm, normal is 400 cubic millimeter. P24 increases. RT-PCR, real-time PCR to detect HIV particles in blood. Value smaller than 5,000 copies per ml of blood is good prognosis. Greater than 1 lakh per ml of blood is a bad prognosis. NAT nucleic acid testing involves amplification and detection of one or more specific target sequences located in specific HIV genes. Detection limit 200 to 400 copies per ml. Human immunodeficiency virus HIV. HIV belongs to the retrovirus groups, RNA containing viruses that replicates with the help of reverse transcriptase, RT or RNA dependent DNA polymerase. Viruses has a cylindrical core containing 2 copies of single stranded RNA. Protein's components are named after its molecular weight. Core contains reverse transcriptase P66 and P55, endonuclease, nucleocapsid P9, proteases P10, major capsid antigen P24, about 200, outer cell myristoylated protein P17, P17 protein having molecular weight 17,000 GP, glycoprotein with molecular weight in kilodalton, GP120, glycoprotein with molecular weight 1,20,000, RT reverse transcriptase, I and integrase, PR protease, S a surface antigen, TM transmembrane protein, and M a membrane antigen, C a capsid antigen. Glycosylation GP120, GP41. Capsid, nucleocapsid, TAT, viral RNA genome integrase, reverse transcriptase, protease, lipid membrane, matrix protein, proteins of host cell, glycoprotein complex, HIV capsid, about 200 copies of P24. Virus entry. The binding of HIV with target cell is through a receptor mechanism. The GP120 of virus envelope which specifically bind with CD4 molecules on the surface of target cell. CD4 acts as a receptor for viruses. The CD4 molecules are present on surface of T helper cells and therefore helper cells receive the maximum attack of HIV. HIV, CD4 and chemokines as antireceptors. Replication cycle. HIV fusion of HIV to the cell host cell surface. HIV RNA, reverse transcriptase, integrase and other viral proteins enter the host cell protein integration complex and then viral DNA is formed by reverse transcription. Viral DNA is transported across the nucleus and integrated into the host DNA. Viral RNA, reverse transcriptase, integrase and viral DNA. Host DNA, new viral RNA. New viral RNA is used as genomic RNA and to make viral proteins. New viral RNA and proteins moves to the cell surface and new mature HIV forms. Virus is released. Viral protease clips new polypeptide proteins to create mature infectious virus. Immunology of AIDS. The CD4 T helper lymphocytes are decreased in number, leading to immunodeficiency. The GP120 surface unit attached with CD4 molecules present on the surface of T helper cells. The HIV preferentially enters into T helper cells and they are lysed. Macrophages and monocytes act as the reservoir of HIV infection, which disseminate the virus to various organs including CNS. In turn, macrophages decreases. T helper CD4 smaller than 400 cubic millimeter of blood. Lymphokines such as interferon, interleukin 2, etc. are lowered. Anti-HIV drugs. 6 classes of anti-HIV drugs were described here. RT, reverse transcriptase inhibitors, integrase inhibitors, protease inhibitors, inter inhibitors. GP120, GP41, integrase, protease, HIV, RNA, capsid, reverse transcriptase. Anti-inhibitors, CD4 receptors, CCR5 or CXCR4 co-receptor, protease inhibitor, integrase inhibitor and reverse transcriptase inhibitor. Reverse transcriptase inhibitors, AZD, dideoxythymidine, azidotymidine, zidovudine, DDI, dideoxyadenosine, didenosine, zidanosine, DDC, dideoxycytidine, gelicitabine, D4T, satavudine, 3TC, lamivudine, abacavir, zidovudine, integrase inhibitors, raltegravir, LVT-gravir and dolutegravir, protease inhibitor, iponiavir, indinavir, nalfinavir, amfaxvir, ritonavir and darunavir and atazanavir. First line choice. Entry inhibitors, maraviroc and enfuvirtide. Prevention. Since there is no cure for AIDS and vaccines are not present, public awareness and education are only means to limit use. Protections. Use disposable syringes. Boiling for 10 minutes will inactivate the virus. Thank you.